Lactic acid bacteria, Lab. Characteristics of lactic acid bacteria. The lactic acid bacteria, Lab, are very effective for improving soil ventilation and for growing fruits and leaf vegetables. Lab are conditionally anaerobic, so they can also survive with oxygen. Lab are resistant to high temperatures. Lab are the strong sterilizer. Lab are used to cultivate IMO number 3. Lab accelerate root growth during transplanting and effectively enhance the initial growth of the plant. Lab increase the solubility of the fertilizer. When Lab is used during the vegetative growth period of fruiting vegetables, higher quality plants will result and may be kept for longer periods in storage. When spraying Lab on a rice paddy field, more weeds will appear. Lab solubilize phosphate in 100-200 ppm. Using Lab in phosphate-accumulated soil will increase its capacity to absorb the insoluble form of phosphate, and help overcome the saline disorder as a result of decomposition of the phosphates. Lactic acid, Lab, can reduce damage from gas through neutralizing ammonia gas produced where the immature compost is applied. Lab have about a week-long resistance to some fungi. There is no significant effect of Lab on damage done by diseases and insects. Lab can adjust the fluids within plants, which may increase disease tolerance or harden their bodies even during rainy season. How to make Lab. Tools. Jar, a big and a small one. Porous paper, paper towel, rubber band. Materials. Rice-washed water. Obtain strong indigenous, pure stocks of lactic acid bacteria in a given region by using rice-washed water. By cultivating massive amounts of bacteria, they can continually be applied to the soil. Milk, unprocessed, if possible. Environments. Optimum temperature is 23 to 25 Celsius. A cool and shaded area with no sunlight. Little change in the environment. Methods. Pour rice-washed water up to 15 to 20 centimeters deep in a jar and cover the mouth of the jar with porous paper, that the rice-washed water must occupy 2/3 the volume of the jar. Lactic acid bacteria populate at 23 to 25 Celsius. 3 to 4 days later, the jar will have 3 divided layers: floating matter, clear liquid and dregs. It starts to emit a sour smell unique to lactic acid bacteria. The separated layer of clear liquid contains the lactic acid bacteria, called Lab pure stock. This liquid is used. Pour milk in the big jar. The ideal ratio of milk and rice water is 10 to 1. Since milk has more nutrients than rice-washed water, the lactic acid bacteria grow vigorously. In 5 to 7 days, at 23 to 25 Celsius, starch, protein and fat float on the surface, called cheese, and light yellow liquid, Lab serum, remains below the cheese. When Lab are produced after 7 days, remove the cheese, which is separated from Lab. If the cheese is left as is, the cheese melts and undergoes a secondary reaction. This cannot be used as Lab. The Lab serum can be kept after filtering and refining. How to keep Lab. Keep the refined Lab serum where there is little temperature change 1-15 Celsius and no sunlight. In order to keep Lab at a normal temperature, it must be mixed with the same amount of brown sugar and stirred with a wooden stick. How to use Lab. The basic dilution ratio is 1 to 1000. If the Lab is used together with mixed compost or IMO, the fermentation process occurs fast, leading to very effective results. Lab function to prevent the fermented mixed compost from decaying. Lab reinforces the ability of anabolism of microbes living on the plant stem and leaf, a condition that arose from the abuse of insecticides and fungicides. When using Lab alone, it is more effective to use it with FPJs. Fields will recover fertility and the soil will become soft and fluffy if IMO mixed with Lab is sprayed on the field. Use Lab 1:500 with FPJ 1:300 as drinking water for livestock to recover their digestive function. Using Lab too much will decrease the sweetness. Lab are extremely effective in making fruits and leaves large, but the amount of Lab used should be reduced approaching the later stages.